Starting up with today's topic, as you can see, it's a very basic. All of you are going to know a lot of things. There will be certain new terms. There will be certain new data that you are going to come across that will be related to the medical field or medical science. Apart from that, you all know what is cell and what are the subcellular organelles. You are aware of it? You all know, right? What should be the proper definition of a cell? My first thing. If anything, we call anything as a basic unit of life, what does it mean? It's the smallest thing? Is it the smallest thing? Please come and say. Fine, basic. Can a smallest thing be functional? My question is. Can the smallest thing act as a functional unit or as a functional house? Just like, some steel plants or something like that. Please come and say. Can it be? So what should be the proper definition that we can provide for cell? Yes, please. Fine. It is a basic unit of life. Fine. Sit down. No need of standing around. It's a basic unit of life. I understood. That maintains or that performs a lot of functions. And that supports structurally also. That has got some structure, right? So cell is the basic structural and functional unit of life. You have to add these two terminologies. Why? Because cell has got a structure. It supports structurally. If we talk about tissues are made up of cell. So tissue has got a shape because of the cell structure. If it is structurally added upon, if added, then it has to be functional. It performs a lot of functions. We are going to see that. Yes or no? Fine. If we classify cell, how we classify cell? What is the classification of cell? Prokaryotes and eukaryotes, right? What is prokaryote? Whenever, this is one of my suggestions that is going to come across, that you are going to face a lot of time. Whenever you feel tough to define anything in biochemistry, just divide that word. That's the easiest thing that you can come across with. Okay? Prokaryotic. Karyotic word has come from carrion. What does carrion means? You know, when we add pro to any name, ancient, no. Primitive, anything else? Which is not clearly identified. In other words, we can say it is not there. Maybe. It can be there, it cannot be there. So we better say not properly identified. That comes as a word termed as pro. Okay. U means for eukaryotes, true. Okay. Karyon means nucleus. So if you have a true nucleus, a visible, identified nucleus, the cell stands to be as eukaryotic. Okay. If no identified nucleus, that comes to be as prokaryotes. Fine. If we talk about a cell has got a structure, and inside the structure we have a lot of other organelles. Can you name some organelles? Anyone? Randomly. Mitochondria, fine. Anything else? Ribosomes, okay. Endoplasmic reticulum are of two types. Rough and smooth, very good. Rough is facing towards nucleus, very good. Smooth is facing towards cytoplasm. Cytosol or cytoplasm. Fine. Anything else? We are leaving, you people are leaving some of the important organelles. Golgi apparatus, fine, very good. Golgi bodies also, some of the books call them as Golgi bodies, fine. Anything else? Vacuoles, okay. Transporting machineries, okay. Anything else? Cytoskeleton, okay. Peroxisomes, very nice. In modern medicine, there are a lot of researches going on in peroxisomes, activity of peroxisomes. Okay, we'll come to that. Anything else? Any very important thing that you have left? Mitochondria हो गया, nucleus हो गया, बिना nucleus के cell का? Organelle I am asking. I am asking about organelle. Have you heard about lysosomes? There are a lot of disorders, lot of diseases that happens because of some lack of enzymes or defect in protein targeting or anything that occurs provided in the lysosome. Lysosome is a very important functional organelle of a cell. Okay, remember these things. Fine. Moving up with the next slide. Why this is not moving? Didi, why this is not moving? Moving. These are the SLOs. SLOs means that is what we are going to come across in our slides or during our 1 hour of class. We are not going to introduce anything about extracellular matrix. That will be in a different part of lecture. Fine. Don't worry. We are going to go through the definition of cell, types of cell, differences between prokaryote and eukaryote, marker of subcellular organelles, a very important topic for your point of view. Structure and function of nucleus, cytoplasmic other components like ER, Golgi apparatus, peroxisomes, mitochondria, and disease associated with subcellular organelles. This is what we are going to focus around. Okay. This is a general story which says or which stays or which conveys us the point that the smallest particle is an atom that conforms a molecule, that forms a molecule. Many molecules form together a organelle. Fine. Composition of many organelle forms together a cell. The cell is the functional part. Composition of cell forms the tissue. Again a composition of organization of tissue forms a organ. Organization of an organ forms a system, organ system, and with that organ system our whole body is supported out. The general life story, a general story of a cell, a journey of cell. The key problem. Okay, this is what you have told. Cell is the smallest structural functional unit of a living organism capable of performing essential vital functions such as metabolism. Point number 1, what is metabolism? Please be loud. फिर से बोलो. Anything, anyone else? Yes, what is metabolism? Okay, nice, fine, very good. In a simpler language, reactions, any reactions, okay. Cycles, any cycles is metabolism. Is it true? No. Metabolism is a process that includes either formation of certain stuff or breakdown of certain stuff. In this way, either they need energy or they produce energy. In the presence of certain enzymes is called as metabolism. Okay? So metabolism can either be anabolism or catabolism. If it is anabolism, it needs energy. If it is catabolism, it is going to provide energy. And all this process is occurring with the presence or in the presence of active enzyme. Is that clear? So entire metabolism that we are going to look around in the entire other topics of biochemistry is properly occurring inside the cell. So cell is responsible for one of the functionality of a cell is metabolism. Okay, growth. How cell grows? Yes. Proliferation provided by which cycle? Anything? G1, G2, S phase, anything? S. What happens in S phase? Replication, doubling of DNA, fine. That causes growth of the cell. Now, how doubling of DNA or replication of DNA makes the cell grow? Think about yourself, think about your body. How it is happening? What a DNA contains? Genes that codes for certain protein? Yes or no? Fine. So if DNA is going to get doubled, the product of DNA is also going to get doubled, yes or no? Yes or no? In this way, as the concentration of substrates, concentration of chemical components, composition of life-giving components increases inside the cell, it grows in size. That happens in S phase, synthetic phase. Is that clear? Once the cell has grown enough, ready for the division, it goes for mito. Is that clear? Movement. Is cell responsible for movement? Yes, of course. Reproduction, I have already discussed about mitosis. Okay. Hereditary transmission. Hereditary transmission is responsibility of DNA. Fine. The hereditary material of our body, of a living organism is DNA. The DNA is carried out by, or provided by parents. Yes or no? Yes or no? Nuclear DNA is provided by? Can anyone? Who provides the nuclear DNA? Father or mother? Father, okay. Who provides the mitochondrial DNA? Mother. Yes or no? How you are going to justify this? Spermatozoa, the sperm has got lot of mitochondria in the tail. In the tail. Fine. But does the tail enters during before the fertilization is about to occur? During the conception, is the tail going to enter? So who carries the mitochondria? It's oocyte, not mother. Mother is not carrying it in hand. It is there in oocyte. Okay? So remember this point. So any mitochondrial inherited disorders, why you guys are late? Train. I was not driving the train. Go inside. So any mitochondrial inherited diseases is the responsible of a mother. Remember this, okay? এটা কোন দিকে দেখালে হয় ভাই? কি রে বাবা? This is a picture of a typical cell that you all have seen. So prokaryotes, as you told, before karyon, before nucleus, unidentified. Okay, remember this point. Lack a well-defined nucleus and possess relative simple structure. They generally, prokaryotes are lower organisms like bacteria and all. Okay. Eukaryotes are true or good nucleus, identified, properly identified nucleus. If it is present, we call it as eukaryotes. Have a membrane enclosed nucleus encapsulating their DNA. Higher organisms, it is generally present in higher organisms like plants and animals. Fine. In this point, if we talk about prokaryotes, where is the DNA then? Where is the DNA provided in bacteria, present in bacteria? Inside. Is there any specific area where it is located? No. It circulates. It keeps on roaming. Okay. But when we talk about eukaryotes, the DNA is properly confined and stabilized and then kept inside the nucleus. Is that clear? If we talk about size, prokaryotic cells are generally lower in size, whereas eukaryotes are bigger in size. You can differentiate. Cell membrane, they have got rigid cell wall, whereas in eukaryotes we don't have cell wall, we have cell membrane or plasma membrane. Cytoplasm is viscous, no subcellular organelles. Eukaryote subcellular organelles are present. You can differentiate. Nucleus is a nuclear zone with the DNA. As I told you, it can keep on circulating inside the prokaryotic cell, whereas in eukaryotes it is present inside a well-defined nucleus. These are all other things that you can look around, like for mitochondria it is absent. For eukaryotes, mitochondria, nucleolus, Golgi body, cytoskeleton, and as well as these all are present. Whereas the cell division goes absence, they generally go for binary fission. In case of eukaryotes, the cell divide provided by mitosis. Very general informations. Yeah. Now coming to a different area where the question arises that How did the researchers, how did the scientists did their study provided on certain organelles? Fine. If I simplify it, how do we come to know? How did we come to know that mitochondria is the powerhouse of cell? We have to do a study on these specific organelles, and these specific organelles are present inside the cell. So what we have to do? We have to take them out from the cell, then put it under put those organelles into certain conditions and studies, and after that we get some result. So the first challenge was that how we are going to take out the organelles out of a cell. That was the first challenge. And the solution provided was with the help of a process or technique called as ultracentrifugation. Have you heard about this term? Common terminology. Ultracentrifugation. A machine which uses centrifugal force provided to that it causes the sedimentation of certain organelles, certain part of the cell, dependent on their density or size. General principle. Okay. This is a technique called as ultracentrifugation which allows the organelles to sediment at certain speed or rate of G or gravitational force provided. Okay. This is the chamber, how it looks like. This is the chamber that how it looks like. You can see there are certain, test tube like holder like components there where we generally are going to keep the homogenized tissues, and we are going to apply force and the motor is going to rotate it. Upon rotation, what is going to happen? See, this is what going to happen. Homogenization is a process where the living tissue is, what is homogenization? There was some instrument in old chemistry labs, rotor and pastel, something like that. मुझे नाम याद नहीं आ रहा. अभी तो we generally homogenize by using some electrical machines, but earlier days we used to homogenize by that rotor and pastel. क्या बोलते हैं उसको? Yeah. Okay. पुराने जब हम लोग जब स्कूल पे थे तो हमारे स्कूल पे केमिस्ट्री लैब पे ये रखा होता था in general. अभी मुझे पता नहीं. I have no idea. So what is homogenization? Very good. Breakdown of anything. Sit down, please don't sit. Breakdown or lysis of a cell. Lysis of cell. That may be a prokaryotic. If it is prokaryotic, then cell wall. If an eukaryotic, then it is plasma membrane. ठीक है? ऐसा नहीं है. This is nothing like that कि you take the tissue, you do motor pestle like this, you keep on grinding, or you have a mixer grinder, take a tissue, put it on mixer grinder and the cell will be separated. No, it is not like that. We need two components. One component is called as twin 20. You can write it TWEEN, twin 20. This is a material, this is a chemical that generally acts as a buffer also, that provides a solvent for the homogenization. Solvent. Okay. That act as a lipid solvent and that allows the lysis of cell membrane without disturbing the integrity of the organelles. So now imagine if you are homogenizing anything and by mistake you have disturbed the entire organelle, structure or integrity of organelle, what will happen? What will be your study then? Zero. You won't get to see the organelles only. So no organelles, no study done. Fine. So for that reason, we add a buffer, we add a solvent, lipid solvent generally, that causes the rupturing of plasma membrane. As a result of that, the organelles or the cytoplasmic components comes out that we are going to separate afterwards. Okay. And this component and the lipid solvent that we add is twin 20. With that, this entire this entire mechanism or this entire process is also done in the presence of one more component that is called as 0.25 sucrose solution. 0.25 sucrose solution. Now the reason behind using sucrose is that sucrose is generally metabolically not active. Cell generally doesn't metabolize much sucrose. Okay, point number one. Second point is the osmotic pressure, or you can say the osmotic challenges that the cell has got inside is same as that of sucrose solution. So osmolytically it is similar, the solution and the cellular component and the cell. Okay, so as a result of that, when the osmolytic pressure is same, the components readily move outside of the cell. That is how we extract things. Is that clear? Is it simplified? We use a lipid solvent to break down the plasma membrane so that the components can come out. Point number one. We are doing this entire process in the presence of another solvent that is called as 0.25 molarity sucrose solution. These two things are needed. Sucrose is why? Because it is metabolically not acceptable mostly. Second point is the osmotic pressure that is exerts is similar to that of the cytoplasm. Okay, so as a result of that, the movement of lot of components from the inside to the exterior part, exterior world or environment becomes easy. That's the only reason. Okay. Next to that, these are the tissue cells we have homogenized. Leading to that homogenization, we have opened up the plasma membrane. Because of that, the organelles are now into the atmosphere, into the environment provided in that test tube. The first thing what we do is, we put that test tube in that ultracentrifuge machine. Okay. We start with 1000 G. G stands for gravitational force. Fine. Specifically for 10 minutes. The first thing that we get is a supernatant and a sediment. Okay. The sediment basically contains the nuclei and some of the cellular debris. Now why nucleus is first removed out? Is sediments. Why? Because of its density. Okay, because of its density. Fine. We collect the supernatant from it, take it into another test tube, and we remove out the sediment and use it for the research purposes. Whatever, if you want to see how the nucleus looks like, what are the nuclear pores like, XYZ. We use the sediment. Okay. But we want something else now. What we use? We use the supernatant and transfer it to another test tube, put it again into ultracentrifugation at 20,000 G for 15 minutes. It keeps on rotating for 15 minutes at a centrifugal force of 20,000 G. Fine. In that part, what we get is in the supernatant, and in the sediment, what we got is mitochondria and chloroplast. If plant cells, chloroplast of course if plant cells. We generally get the mitochondria in this situation. Fine. Again you got two things. You got the supernatant, you got the sediment. If you want to study for mitochondrial, if you want to go for mitochondrial studies, what do? We collect the sediment, we go for the study. If you want something else, we are taking out the supernatant, transferring it to another test tube, going for ultracentrifugation, but at this time it is at a higher speed rate. It is at higher speed rate provided to 1 lakh centrifugal force at 1 lakh G, provided for 60 minutes. We keep on rotating. We keep on rotating the supernatant. At this moment of time, we got pellets containing microsomes. What is microsomes? This is an interesting twist that we are going to see during differential centrifugation. What is microsomes? Anyone? You all have come across with these terms and terminologies. I hope so. Because हमको लगता है कि सब कोई तुम लोग कोचिंग वगैरह से आए ही होगे. You all are okay with sometime I use Hindi. You're okay? Because there are some outstate persons, so that's why I'm using Hindi. What is microsomes? This is not expected. Anyone? Any guesses? Fine. Huh? See, it is endoplasmic reticulum. The twist over here is when we were doing the process of homogenization, sometimes the endoplasmic reticulum gets ruptured, okay, because of their spatial arrangement and their membrane and their, the structure they have. They get ruptured, they get disturbed, their structure is disturbed or ruptured. So the debris of endoplasmic reticulum provided during the homogenization is nothing but microsomes. Is that clear? Fine. So we got that. Again, if we need to do any study about the endoplasmic reticulum, then we collect the debris, we collect the pellets. If not, then we transfer the supernatant to another one, go for 1,50,000 G, and for 3 hours we get ribosomes provided in the debris or the pellet. This is how the organelles are segregated. Fine? Is that clear? There is one more technique, isopycnic centrifugation technique, that generally is dependent again on density based, and the solvent that we are going to use is again sucrose solution. The reason I have told already. But in this case, in isopycnic centrifugation technique, we don't use 2 in 20. Is that clear? Is that clear? Interesting thing about here is, this picture is going to be very important for you in terms of examinations and questions. Remember that. It is getting recorded. It will be delivered to you. Don't worry. Is the picture clear to you? Is the process clear to you? Fine. Now the next point comes across is, I told that we are going to separate the organelles, we are going to study the organelles. Fine. Can we see the organelles through our naked eyes? Can we see the organelles specifically? Can we see the organelles specifically under microscope? We need an electron microscope. Not available everywhere. Fine. So how you come to know that, okay, fine, I have done this ultracentrifugation, I have got this debris contains nucleus. This debris contains mitochondria. How you know? Very nice. Okay, sit down. We come to know about the details about it provided by the presence of certain enzymes. Okay, so all organelles have got their specific enzymes, and we call those enzymes as marker enzyme. The second important question coming up that is going to come up for you regarding this topic. What is marker enzyme? Fine. Marker enzymes, the name suggests marker enzyme that marks the presence of something is called as marker enzyme. So all organelles have their own marker enzymes. Like, is that clear? For example, sorry. See, if we talk about nucleus, lamine A and C, these are proteins, fine. We can identify them provided by the presence of lamine A or C. In other case also, if you ask me a specific enzyme only, then I may answer like if the debris is nucleus, then the enzyme that has to be present inside the nucleus is DNA polymerase or RNA polymerase. Okay? Marker enzyme. So you got to know that yes, I have got DNA polymerase in this debris, then what does it mean? The debris is nucleus. You are confirmed. Fine. If it is mitochondria, if the debris has got mitochondrial inner membrane, the enzyme is ATP synthetase. The complex 5. The complex 5 of electron transport chain, right? Mitochondria outer membrane, we have monoamine oxidase, same like that. You should remember all these names. Fine, you have to remember. If it is mitochondrial matrix, we have got citrate synthase. If it is lysosome, we have cathepsins. If it is Golgi bodies, galactosyl transferase, microsomes, glucose 6-phosphatase, cytoplasm, lactate dehydrogenase, and perioxysomes, catalases. These are the enzymes that you are supposed to remember, because this is important question that is very frequently they will ask you. Now, what is marker enzyme? Name the marker enzyme of mitochondria or name the microorganisms of lysosomes. Okay, please remember that. You know, in all this thing, this microsome, what is microsome? Endoplasmic reticulum, fine. This enzyme has got a very important role in carbohydrate metabolism. Remember this enzyme's name, glucose 6-phosphatase. If there is a deficiency of this enzyme, that is glucose 6-phosphatase occurs, it causes a very lethal disorder that is named as von Guericke's disease. Okay, you are going to see this enzyme properly during your glycogen metabolism. Remember that, fine. And that enzyme is specifically present in, if you ask me the localization of this enzyme, it is present over the smooth endoplasmic reticulum, but not in rough endoplasmic reticulum. Please make it clear. Okay. Moving to the next slide. Let's talk about the organelles a bit. I hope you people know this is the structure of nucleus, where we have nuclear envelope, we have chromatin inside, condensed chromatin, we have nucleolus, we have nuclear pores, and we have cisternes. Fine, cisternes are present over there in endoplasmic reticulum, rough endoplasmic reticulum. Fine, it is not a part of nucleus. What is nucleolus? A very common question. What is nucleolus? What does nucleolus holds? Very good. It holds the RNA. Are you aware of it? Then bola kyun nahi? Nucleolus holds RNA. The majority of the RNA are stored over there in the nucleus. Fine. How chromatin is condensed? How the DNAs are stocked and packed? Provided in the nucleus? I still remember it was given in my 12th class biology book. A very beautiful picture was given. Histone, yes. Abhi bhi yaad hai mere ko. Octamer, yeah. How DNA is packed? Big coil of DNA, 2 meters of DNA specifically in each cell. How it is packed? They are packed up first with histone proteins. Fine. This histone protein causes the condensation that converts them into chromatin. Further, the condensation occurs, providing the chromatin to convert into chromosomes. Yes or no? Fine. General story about nucleus. It is present in all eukaryotic cell that can divide. Has 23 pairs of chromosome which contains DNA originally inherited from each parent. Nothing new to. DNA is 2 meters long, but it's compressed. As I told, most nucleus has a nucleolus patch which is rich in RNA. Point to be noted, nucleolus does not contains only RNA. It has got some proteins also. Some targeting enzymes, some protein targeting enzymes, or some enzymes that are responsible for the transcription process to occur or for the RNA to get synthesized. Okay, it also contains. Nucleus is surrounded by the nuclear membrane. It is a double layered membrane. The inner layer is generally in contact with the nucleus. We call it as perinuclear membrane. Fine, we call it as perinuclear membrane. And the external membrane is in contact with endoplasmic reticulum. Is that clear? Apart from that, the nucleus have also got nuclear pores provided in the inner membrane or the perinuclear membrane. Now, what is the role of nuclear pores? Please, no need of standing. Baith ke bolo. Acha, my question is, fine, okay. Is nuclear pores responsible for entry of substrates or exit of substrates? Somebody said exit. Both. Okay. It is not only responsible for the exit of substrates. It is also responsible for entry of substrates provided for certain targeted molecules. Fine. It is not permeable to everything. It is selective permeable. Okay. Is that clear? Nuclear pores is the provided reason, the provided tags or protein or molecular proteins, okay, that are responsible for the exit of a matured mRNA. Is that clear? This is how the under electron microscope the nucleus looks alike. It is as if it is sitting as a crown, you know, over the hat, and the hat appearance is nothing but endoplasmic reticulum. This is more close picture of a nucleus. This is how a histone the DNA is packed in the, with the help of a histone protein. These are the electron microscope pictures. You know. Yeah, here is a clear picture where you can see a strand of mRNA coming out of a nuclear pore complex. Well, I have told you about nuclear pore. Nuclear pore is a pathway where the things can go inside or things can come out of it. Now, what is nuclear pore complex? Anyone? What is nuclear pore complex? Very simple. Anyone? जोर से बोलो. I never scold, don't worry. Protein pump. Anything else? When we call anything as complex, it has got a different structure. No. It's looking same, see? Nuclear pore complexes. What makes nuclear pore convert into nuclear pore complexes? That is what? Target molecules. Signals. What are signals? Proteins, peptides. Fine. So, nuclear pore complex is a combination of nuclear pore with certain signal. Fine. That signal can be extranucleus or intranucleus. Fine. So, this complexes together allows, it gives the signal provided for the exit or the entry of any of the components. So, that is why here it is written as nuclear pore complex, because you can see it is showing you the exit of an mRNA. So, that is only possible with a signal. Signal can be a peptide, a protein or anything. Okay. Coming to the ER, this is the diagram. The smooth endoplasmic reticulum facing up the cytoplasm, whereas the rough endoplasmic reticulum is added or adjacent to the nucleus. Okay. Oops. Very simplified. It is a complex of series of tubules inside the cell, chain of tubules, all lying in a very unstaggered manner. Fine, there is no proper positioning, no proper hierarchy. It is all aligned randomly. Fine. And these are tubules. They have spaces through which components can move across. You can see here. See, these are the spaces. These are this beneath this. These are the spaces where the components can move across. Where are the components coming from? They are coming from the nucleus. So, components coming up from nucleus moves into the path or patches of tubules of the endoplasmic reticulum and finally leaves the endoplasmic reticulum from the smooth endoplasmic reticulum, or sometimes directly from the rough endoplasmic reticulum. Okay. What is rough endoplasmic reticulum responsible for? Synthesis of. So, who is going to leave rough endoplasmic reticulum? Matured protein or immatured protein? Immatured protein. Okay, remember this, immature proteins. One end is continuous with the nuclear membrane, two types of ER, that is smooth and rough, you all know. Rough endoplasmic reticulum is responsible for synthesis of proteins. Okay. Initial folding of proteins, of course. Smooth endoplasmic reticulum has also got some importance. First of all, it has got a very important role in cholesterol metabolism. In not all the cells, but some specific cells which are responsible for synthesis of cholesterol as a steroid. Fine. Why I am saying this? When you will go through the cholesterol metabolism, you will see there is a rate limiting step of cholesterol metabolism. Okay. I don't want to rename the enzyme and all. That enzyme is specifically located on the smooth endoplasmic reticulum. Is that clear? It is located on the smooth endoplasmic reticulum. So till now I have given you two important points regarding smooth endoplasmic reticulum. The first one was marker enzyme, microsomes. Kya tha? What it was? Glucose 6. Very good. You have to remember that. Second important point, steroid synthesis, basically cholesterol metabolism. Okay. Third important point about smooth endoplasmic reticulum, it is responsible for lot of detoxification reactions, specifically like drugs called as aminopyrine, morphine. These all drugs are detoxified provided in liver in smooth endoplasmic reticulum that has got cytochrome P450 microsomal degradation system working on it. What is that? What is that? Repeat it at least. What did I say? Smooth endoplasmic reticulum of hepatocytes has got cytochrome P450 microsomal degradation system working on it, that is responsible for detoxification process of various drugs. These drugs that I named, like morphine for example, there are many other drugs. Okay. So third function of smooth endoplasmic reticulum is detoxification. The last one I have already told you. Fine. Generally आप books search करेंगे, आपको smooth endoplasmic reticulum के functions बहुत कम देखने को मिलेंगे. You will get to see functions of smooth endoplasmic reticulum very less. But medically it is very important. Okay. Physiologically it is very important. Ribosomes, 60S, 40S subunits, we all know, we have gone through translation many more times. We have seen how ribosome does it, what ribosome has got inside. The larger unit has got 3 sites. What is that sites? Exit site, peptidal site, amino acid, tRNA site. Yes or no? Fine. Major function of ribosome is to responsible for translation, synthesis of proteins. Okay. Where is ribosome present? Is it free? Is it circulated free or it is attached with something? Does mitochondria has ribosomes? Yes or no? Are they similar to that of these ribosomes? Very good. No, they are totally different. Fine. They are membrane bound. Generally synthesis of membrane, like membrane bound organelles like Golgi apparatus, lysosomes, endosomes or secretory proteins. Basically the overall role of ribosome is nothing but it is responsible for synthesis of proteins. Mature protein, immature protein. Immature protein. Okay. This is what peptidal synthesis that we talked. Next is Golgi apparatus, flat stacked upon structures. Okay. And the function of Golgi apparatus is what? Protein receiving, protein processing, packaging, targeting. ये 4 points याद रखिए. पहला, protein processing. I told you that when the protein is getting synthesized, coming out of the endoplasmic reticulum, it is immature. So the maturity takes place at, okay. Now all the proteins are to be, have got a signal, have got a target, organelle or target area. XYZ protein is produced inside the cell, it has to move to XYZ organelle. Let us say it has to move to lysosome, for example. Someone has to take it away, no? That process is also done provided in Golgi bodies. That is what we call as protein targeting. There is a separate class provided for protein targeting. Okay. Apart from that, packaging and how these proteins are going to go. They are going to move with the help of vesicles or vacuoles. This packaging is provided by Golgi bodies. Okay. It has got 3 parts: cis, mid and trans. The protein is first received at the cis part. All the processing, all the packaging, all the targeting, all the modification that we call as post-translational modifications and all is done at medial part. The exit is done provided by transpart. Simple. Okay. This is how it looks like. This is the process, entire process of receiving a protein. You can see it has received the protein with the help of transport vesicle. It enters the Golgi body, sorry, the Golgi apparatus. It has got a cis phase where it is received, medial phase where all the processing is done, trans phase where it removes in the form of a vacuole. If it is going outside, for example, like insulin, beta cell of pancreas produces insulin, but action of insulin is inside beta cell? No, outside the beta cell. It has to be packed in certain vesicle that goes and attaches to the plasma membrane and causes exocytosis. Those type of vesicles are called as secretory vesicles. Okay. This is all where these proteins that have been matured, that have been provided, that have been packed well are going to be. They may pass through the plasma membrane to the surrounding medium. Point number one. They may reach the plasma membrane and integrate themselves. The plasma membrane has got some proteins, transmembrane protein, integral proteins, yes or no? They may integrate themselves into the plasma membrane. They can act as integral or membranal proteins. They formed a secretory vesicle. Just now I told you about insulin. I gave an example of insulin. Fine. They can be, the synthesized material may be collected into the lysosome packets. Acha. Now, this point is quite vital about to know that what is the role of lysosomes. So that's why I added this point to the last part. Okay. This is lysosome, a very important structure. Golgi apparatus is known for packaging, processing, shipping, targeting of proteins. Lysosome is known for digestion. Digestion is not a proper word. Phagocytosis, okay. Autophagy, fine, okay. Anything else? Destruction. Of which sort of proteins? Are all proteins going to get destructed? No. All proteins have got a life, span of life. Once the span of life is completed, or any protein that is mutated, that has got mutated, whose responsibility it is to degrade those proteins and reutilize them for the cell purpose? Lysosome. What lysosome has got? Very nice. Both of you are correct. But when you talk about digestive enzymes, on the other part you say hydrolytic enzymes, remember one thing. Very soon you are going to go through enzyme classification. IUPAC enzyme classification. There will be a classification called as hydrolases, sorry. Okay. Remember that all digestive enzymes are hydrolytic by nature. Okay. If we say that hydrolases or hydrolytic enzyme, what is the meaning? It requires water molecule to break certain component. Simple. So, lysosomes are rich in acid hydrolases. What? The pH of lysosome is near about 5. Acidic? Fine. So, all enzymes that are responsible for digestion or degradation of a immature protein or mutated protein are provided in lysosomes. Is that clear? Sorry. Is it only proteins that gets broken down? No. No way. It can be carbohydrate, it can be lipids, it can be nucleic acid or nucleotides, or it can be a protein. So overall, the role of lysosome is just like an area of a city where we generally put our garbage. They destroy the garbage with certain things and reuse. होता है ना वो बड़े-बड़े शहरों में मैं एक बार दिल्ली गया था वहाँ पे देखा था मैंने. What they do? The old vehicles, you know, they have got scrapped vehicles. They remove the entire parts of scrap vehicle, they keep it in somewhere else. And they use that scrap materials for production of certain other things, useful things. That is the role of lysosomes. Okay. These are the enzymes. If we talk about degradation of carbohydrate, they have got certain enzymes like glucosidases. These are all hydrolytic enzymes, remember. Glycosidase, mannosidases, glucuronidase, hyaluronidase, aryl sulfatase, protein hydrolyzing enzyme like catepsins, pepsidases, elastases, collagenases, ribonucleases and deoxyribose. These are all basically, these are all enzymes that are hydrolytic by nature and responsible for the degradation of macromolecules. That can be carbohydrate, that can be lipid, that can be proteins, that can be nucleic acid. Now here comes the important part of the story. Lysosomal disorders, very important. Urate crystals. Many one of you must have heard about a problem called as gout. Severe pain provided in the lower joints. I have written it as, I have shown it as, the top of the toe, but it also sometimes occurs in the knee. Gout. What happens? In gout, this type of crystals, these are uric acid crystals, urate crystals, they start getting accumulated inside the cell. Fine. Is that clear? And as lysosomes, they start phagocytosing or phagocytose those urate crystals, causing accumulation of crystals inside the lysosomes. Okay? Result of that, which causes a physical damage to the lysosomes, and after all, the lysosome enzymes, acid hydrolases are released inside the cell. That causes autolysis of the cell, responsible for the severe pain. That is what the responsible of severe pain. Fine. That also causes inflammation, and as a result, arthritis occurs. Many of you must have seen in TV or pictures, or like, when the dead body is left out for a longer period of time, they generally get a different coloration. Fine. That is due to, following cell death, the lysosomes ruptures. Lysosomes ruptures. The rupturing of lysosome causes the release of acid hydrolases or hydrolyzing enzymes, digestive enzymes, tissue digestive enzymes into the surrounding, leading to you can read, postmortem autolysis. Autolysis is what? Self-destruction. Okay, fine. Next is, cathepsins are implicated in tumor metastasis. Important. These are normally generally restricted into the interiors of, these are proteins, they are normally restricted inside the lysosomes. But in certain cancer cells, they liberate cathepsins out of the cell. That causes the degradation of basal lamina of the cell, leading to hydrolyzing of the collagen and ultimately leading to the tumor cells to travel out of the neighboring area. That can lead to metastasis. So this is how a cancer cell travels out of the border and goes into different area of the body or of the living organism. That causes metastasis. That is provided due to cathepsin. Now cathepsin is inside the lysosomes, but due to some damage, due to some cancerous activity or anything, the lysosome releases cathepsin that comes out and destroys the collagen and elastin. So once the collagen and elastin is destroyed, the basal lamella, the supporting background or the extracellular matrix is removed, leading or helping the cancerous cell to travel into nearby areas. There are some other lysosomal disorders like Fabry's disease, Tay-Sachs, Gaucher's. In other terminology, if you remember these lysosomal disorders, you will read this lysosomal disorders in detail, each of things. And they will be named as lipid storage disorders. In some architecture, in some articles or literature, you may find the names of these diseases as under lysosomal storage disorders. Basically, it is nothing, but this is due to the deficiency of certain enzymes that may lead to these disorders. Okay. This is another disease called as eye cell disease. Basically, it is due to improper protein targeting Generally, some enzymes that has to go to lysosome has a lacking of mannose-6-phosphate, that is a signal, that is a target. So when there is no target, it cannot move inside the lysosomes, it keeps on traveling in the cytoplasm, and itself it is a hydrolytic enzyme, so it starts causing autolysis or degradation, leading to eye disease or eye cell disease. Silicosis, that is responsible again for the scarring of lungs or infarction of the lung tissues provided due to deposition of silica. When we inhale a lot of silica, causes a scarring of tissue and ultimately leading to decreased lungs elasticity. Peroxisomes, a very important organelle responsible for metabolism or catabolism, or you can say the degradation of lot of important free radicals, because peroxisomes are rich in two enzymes called as peroxidases and catalases. These enzymes are responsible for degradation of free radicals or converting the free radicals into acceptable forms. The most commonly targeted component provided for formation of a free radical is PUFA, polyunsaturated fatty acids, that are present in the membrane. Is a common easiest target for the free radicals that may cause rupture of the cell membrane, rupture of the membrane enclosing the peroxisome, leading to the death of the peroxisome, you can call it like. As a result of it, it may lead to some other disorders, many other disorders. Okay. These are some ALD, adrenoleukodystrophy. Again, Zellweger syndrome is again due to improper protein targeting. Primary hypoxaluria again due to improper protein targeting. The enzyme name is alanine glycosylate aminotransferase. Fine. It has not got a proper target. As a result of that, this enzyme is unable to move into the peroxisomes. Fine. As a result, this enzyme is not going into peroxisome. It is causing an improper metabolism of a carbohydrate component called as glycosylate, leading to excessive accumulation of glycosylate. Okay. Mitochondria, powerhouse of the cell. I'm hurrying up little because I'm already running out of time. I have 5 minutes left. Is there anything new about these points you don't know? Is there anything new? Mitochondria is responsible for production of energy in the form of ATP. Major cycle, TCA. Apart from that, urea cycle. Is urea cycle completely occurs in mitochondria? Partially in cytoplasm, partially in mitochondria. Okay. Basically, if you see, mitochondria is a house with lot of enzymes present in their matrix, which causes the complete digestion or complete metabolism or complete catabolism of carbohydrate and lipids provided into water and CO2. Is that clear? Apart from that, mitochondria has two membranes, inner membrane and outer membrane. Outer membrane is generally permeable, but inner membrane is selectively permeable. Okay? Remember this point, is selectively permeable. It doesn't allow all the things. It has to allow a selective components. Most importantly, the inner membrane of mitochondria has got electron transport chain or the complexes of electron transport chain, except complex 2. Complex 2 is not embedded in the inner membrane. Rather, it is hanging just like a fan from the inner membrane or the inner surface of the inner membrane. Okay? It also houses, what is the marker enzyme for mitochondria? If it is inner membrane? ATP? Remember it. Okay. This is already I have told. One last thing that I want to conclude with you guys is attendance. One last thing that I want to conclude you about mitochondria, and another thing, a very important, clinically very important, that is called as ER stress, endoplasmic reticulum stress, ER stress. Pathogenically, it has now coming up into, the studies that even including type 2 diabetes is one of the pathogenesis responsible of ER stress, endoplasmic reticulum stress. Now, what is endoplasmic reticulum stress is, endoplasmic reticulum is rough endoplasmic reticulum responsible for production of certain proteins. Fine, we all know. So, when there is an overload over the endoplasmic reticulum for production of a certain proteins in a over demand or it is unable to supply it, there is over demand of it. So, the endoplasmic reticulum falls under stress. It goes under stress. That causes lot of other pathogenesis. So, basically ER stress, if we talk about, just take an example of diabetes mellitus type 2, that happens because of the resistance to insulin. Now, because the body, the peripheral tissue is resistant to insulin, the beta cell of pancreas is producing lot of proinsulin. So, the functionality of endoplasmic reticulum is increased. That causes endoplasmic stress. That is also responsible for many other pathogenesis or many other diseases to happen again together with. Okay, you will read that in detail in later classes, ER stress, but remember this point. Okay.